Good afternoon, biology students. It's Monday, March 23rd in our week 2 of biology at Hope Academy Online. Today's learning target is going to be to know how viruses are spread in a host and the methods of innate immunity present in our body. So first, before going into anything else, I'd like you to do the entry task. Just list something in the response to this video that you have heard about COVID-19 in news or in class, anything at all. Respond to the video thread with one line, or remember not to respond to somebody else's. This will be your record of attendance. Go ahead and do that right now. As you return, a few things here just about class. I want you to read the section 1018 and 24.1. 1018 is a brief look back on RNA and genetic material in viruses. 24.1 is the beginning of the immune system, looking at the human body systems and organ systems that we're focusing on here in this section. We'll have a short quest on Thursday on the 12 organ systems and animal structure and function. This is in these sections here, we focused on a couple of different things in there, particularly on the general functions of these 12. That was part of your homework that was due today, as well as the organization going from cells to tissues to organ systems. So I'll give a little more information on that tomorrow. If you have questions, you can let me know. A short quest we'll do as a quiz that will have a certain number of time on it that we'll do at the very end of our class on Thursday. So our first academic achievement here in digitally that we have there. Just another reminder too to make sure you're keeping up with assignments. If you're missing something from last week, let me know. Check on the assignments tab in the team site, and you can see how you've done. I put those into iNow as well. Also, the test I do have done, I put that in there last night. If you have questions about your test or you want to look over that, I'm going to post a video once everybody has taken it. I'm working with a few to get that done who were missing in class in the crazy week of Thursday before we left for the long weekend, and then only coming to school one more day. Once I get that done, I can send you out a scanned copy that you could correct and take a retake if you want. A couple of notes on it I'll explain, but basically everybody got a 2-point bonus, kind of call it the COVID bonus, I guess, but just with one question on there that I think was misinterpreted, and the fact that it really was a unique and a bit extra stressful in taking it on that Thursday, and so that's reflected in your score. Your raw score was just added 2 points to it. And I'll give more in that video that specifically goes through those test questions. So back to our task for today, to know how viruses are spread in a host and how the methods of innate immunity are present in our body. Part of this is to be able to get at really what's going on with COVID-19, and this is constantly changing, but I think it's a great opportunity to see some of the things we just finished learning about in our genetics unit and how it plays out in very, very real life as we all are dealing with this right now. And one is just there's a lot of misinformation, and two, there's a lot of work that's being done right now that requires a level of understanding that we started with in this last unit. And so I want to just briefly go over what's happening during a viral infection. This is a general one from your book in section 1018 that I want you to look over. A couple of specific things here in COVID that I can point out to, but in this is just showing you how a virus itself ends up going into a cell and causing damage. We'll learn more about that here this week, but specifically with this example. So let's say this virus is this is a general one, but let's say this is the COVID-19, the SARS type virus that was identified. A few things to note is that viruses all have something unique on their surface right here. This glycoprotein spike. It's some aspect of their protein coat that is specific to them that helps them latch on to cells. And these cells could be and usually are for particular animals because they're very genetic specific. In the case of COVID, this is called, not COVID itself, but COVID latches onto a protein called ACE2. Okay, that's the way that it enters into the cell. This glycoprotein specifically latches to this protein on our cell surfaces that has an exact match. And this is generally where the mutations take place. This virus, we believe, as far as the studies that I've read, was in a snake or a bat. There are viruses that affect all living things. There's viruses that affect bacteria, that affect fungi. They're all over the place. And they're this in-between living and non-living because by themselves, they just sit there and they don't do anything until they find a living host. When this virus then is able to latch onto a cell, and this is most likely what happened, that there was some type of mutation in this glycoprotein right here. So in the genetic material, there was a switch that happened and now allowed it to latch onto this particular protein in humans. And when that happened, that virus is able to latch on, and what it does here is it melds its membrane with that of the host and just spills its genetic information right here. So this is RNA. Most viruses, or many of them at least, are RNA based. And RNA, if you remember, is a single strand of genetic material. And it's the same thing. It's our A and T and C and G, except for the T has been replaced with a U. So the RNA genome for COVID-19 was actually just recently determined. You can go to the same website you did your project on and find the exact sequence of the COVID-19 viral strand. And it's about 30,000 base pairs long, and it's single-stranded. And this is key to being able to both understand it and to identify it. So what happens when this enters into the cell, essentially this instructions is not a huge set of information. Remember, ours is about 3 billion base pairs. This is 30,000, orders of magnitude less. Its main job is to create more of itself. So it's going to hijack our ribosome. So protein synthesis, remember, takes place at the ribosome. It hijacks that system, and it begins to make its own proteins. And once it makes its own proteins, it will then reassemble a new virus, which then emerges and goes and repeats the process again. So a few things in here that are important that are going on right now, and I want to emphasize when I say in class things like we need people to go into this, this is the exact reason why, is that we need people to be working on this at all different levels, public and private, to figure out what's going on. A couple of things that have already happened in the past couple of months is one, they've identified right here the strands, the particular base pairs of this genome. What that allows them to do is that basically what they will do is they'll take, if this is my RNA strand, they now know a way and they can cut this up into smaller segments, and they can amplify it and make lots of it in a sample. And then they can find a different, a matching corresponding pair, and they can identify that in a sample. So if, let's just say that there's a letters here in my base pairs of like U, T, T, sorry, not U, T, T, U, 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 A, C, G. If that's a portion of this genome, you'd want a lot longer one because the chances of something else having this as well would not be very far off. So usually this is many hundreds of base pairs, but let's just say it's just this right here. They'll develop a test that is able to say, okay, I'm going to go around and look for in all these different pieces of genetic material from a sample, let's say from some saliva, where do I find, or do I find this specific sequence that I know came from COVID-19? And when it does find it, it will latch onto it, it will connect to it, and it will then show some kind of signal. Usually it will glow. They'll put some kind of phosphorescence or maybe a other type of marker on it, but they'll be able to tell, look, we found this specific sequence of nucleotides that is only present in COVID-19. And that's the test right now. They're really working on getting ramped up. There's a version it's been used in South Korea and in Europe, and it's a big area that we're trying to buy some time to get more people tested to really know who has this in the population and to be able to better control it. So that takes some knowledge, it takes some work to make sure that that can happen. The second thing then is going more in detail that we don't know yet is how exactly does this 30,000 base pairs, what do they do? What are these particular proteins that they're making? What are they doing? And how can they be affected? And so there's a vaccine, we'll talk more about that this week, about that that is being worked on. But how does this virus do its thing? What particularly are these genes coding for? And how could they be manipulated or turned off or attacked? And there's a lot of work that goes into this. So right now there's people in genetics and in medicine that are working on this right now. A couple personal notes. I have my older sister is a doctor at Mayo, and I have my wife's cousin is a doctor is a genetics researcher in Texas, and both of them have essentially said the exact same thing in the news, is that there's a lot of unknowns about this. And while we hope that this is an overreaction, right now this is not an overreaction. Right now we're trying to get enough time so we don't overwhelm our hospitals, and we can understand this better and get a better sense of who actually has it in these cities. Because most places will say that in Minnesota, as of right now, I think we have 137 cases. Pretty much everybody who works in this would say we have many more than that, but we just don't know because we're not testing enough. And so we're working on that. Once that's figured out, then we can have a better sense of how do we handle it? What are some different things that might work on this right now? Another thing to note is that a big part of viruses are mutations. Is that you know that a mutation is when a letter that say it should have been in the genome AUCG, if this becomes AUC and this turns into another A again, right? If that mutation happens, well, now you have different information that can change the protein. And it might not do anything, but it might cause something big. And in this virus, And viruses in general, they don't have systems in place to help correct mistakes. If you remember in our DNA strand, we've got two strands. So we've got A matches with T and C matches with G. And so when they are copying in that big long strand that we worked on in class in replication, there are systems in place to go make sure that they match them up the right way. And we have very few mutations. Viruses don't have that system. So they sometimes mutate at over 10 times the rate as a typical cell. This creates a lot more opportunity for them to change and to have some kind of new adaptation to their environment. It also has the possibility, as happened in many cases, most recently Ebola, is that there's mutations that all of a sudden what made this virulent for people now just gets turned off and mutated away from being harmful to people. We just don't know. So there's a lot that's being discovered and worked out with this right now. Still in the learning phases, we know a lot, but there's a lot left to do, which is another reminder to wait, follow all the precautions of social distancing and things that you've heard so much about. This is part of the reason why. Okay, so we want to get more in detail of the immune system and how that works. Today, we're just going to be looking at what's called innate immunity. Innate just means inside of you already, and it's a general type of immunity that does not depend on any specific type of infection. So this is always present in our bodies, a few different things that are going on. And so pathogens is a general term for all things that cause disease, viruses and bacteria, which is our two big categories of pathogens. There are other things like there are other small organisms, protozoa, that would fit in here as well. But this is the main thing that we are facing that cause the largest amounts of human impact would be viruses and bacteria. And our immune system that's the body system, one of our 12, that actually causes, that helps to fight against these causes of disease. And innate immunity is right there right away, so it acts immediately. And they're the same no matter what, whether you've seen this thing or not. And we're going to look more specifically what we do when we've seen something before, but this is just a general defense system that we all have. A few different terms here. In this process, we've got a lot of different vocab, so I'll be read through that here and look at those, and we'll look at a lot of vocab, and I'll kind of specify. For right now, I just want you to read through this section and see the overall work of it. But it does include things that are general barriers, so physical barriers, so skin, mucous membranes, those are the things that just keep things out, and also house our own bacteria that help keep things, keep it free from invaders. And then these other series of things that go on here that I'll get to in just a moment. All right, so you've got external. If that gets bridged, if you get a cut, if you inhale something that doesn't get caught in your mucus, or that you don't cough out, or if you swallow something that doesn't get destroyed by your stomach acid, if it reaches inside of our body, then we have internal defenses. So we've got these series of things here. So external is like the wall on the outside, and then internal are like the ways in which we go after it once it's inside of our system, right? And one type that we see here is we see neutrophils and macrophages. These are both different types of systems that we have in order to fight against invaders by just swallowing them up. So this is an example of what a macrophage does. And a macrophage will sense a bacteria or a virus, and they'll engulf it in this vacuole. It's a term we learned in cells when we're looking at cell parts. And different enzymes in a lysosome will connect to it and will just chew it up. These are enzymes that will attack lipids, they'll attack proteins. They're specifically designed to break apart foreign things. And these little pieces right here, this is a mix of whatever's left over. So these are my biomolecules. These are like individual amino acids that have already been taken apart. These are now some sugars, probably some monomers, individual sugars that have been pulled apart there, and also our individual nucleotides. These can be recycled and used again now by the body. They've been kind of torn apart. The machine doesn't work anymore, and they've been worked there. So macrophage is one of those things that works in that system. We also have interferons, which are proteins that work with getting rid of viral infections. So they're just generally present and get rid of a lot of viruses that we just see on their own. And why exactly does COVID cause a problem where others just get taken care of is also part of the question. What's not working as well here is being trying to be understood a bit better. And then we have things like natural killer cells, which probably one of my favorite names in biology. They recognize and help kill cancer cells and virus-infected cells. As their name implies, their job is to, by their nature, kill things. We also have a complement system. A complement system is a group of proteins that act together with other defense mechanisms. They are many number of them that kind of go in a cascade or a series of different responses to help identify and then to attack those things as they come. And finally, I want to end as you read this here and want to focus your attentions on getting the reading done and to look over your 12 different organ systems. But as you look more at the COVID virus, but just generally speaking, our immune system is the question is how do they recognize pathogens? So not just phagocytes, that's the question here out of your book, but in general here, does our whole immune system recognize what is ours? So I call this kind of the us versus not us. How do they recognize that cell A is, hey, that's part of my skin, and cell B that comes in contact, well, that's part of the enemy, that's a bacterial infection, I want to get rid of it. There's a whole host of ways this is done, and sometimes when this doesn't work correctly, or when it gets kind of tricked by a virus or bacteria, it can get our own immune system to attack our cells, because our immune system is actually quite efficient at destroying cells, which is great when we want those cells destroyed. It's not great when there's something that we want there. One quick example before we head out of this video would be diabetes. We've talked about this in a number of different places, but if you know anybody with diabetes, it is ultimately an immune response. It's an immune response that has gone awry, that has actually gone after the insulin-producing cells in our own body. Our immune system has attacked our own body and destroyed the cells that make insulin. Why exactly it does that is still being understood, but it's an example of where immunity is great and we need it, but it can be turned and twisted in ways that will cause problems as well. And we're still looking to understand more why that happens. So as you finish this section, these two sections of reading, look forward and and prepare for our short quest on Thursday. That'll be an online at the end of the time that you'll take in a set of time, probably about a 15-minute period of time here on Thursday. Any questions or other comments, you can put them in the questions section of our page. Have a good rest of your day, and make sure to be ready for that exit ticket when it comes on the screen as well. Talk to you later. Bye.